2020, and we are working on lecture unit 4, Metabolism. This is the second PowerPoint, the Metabolism Part B PowerPoint, and I'll try to break it up again maybe into 10 to 15 minute sections. All right, so again, I always have some challenges with my mouse. Okay. So most of the PowerPoint will be on ATP production by chemoheterotrophs, and you'll recall chemoheterotrophs are organisms that require preformed organic molecules as a source of energy and carbon. So for example, humans are chemoheterotrophs, and you might recall that cellular microbes that invade another organism and cause harm, they too are chemoheterotrophs. The reason they're invading the other organism is that that organism represents a source of preformed organic molecules. So the processes we'll be describing, beginning with the most primitive way of making ATP is glycolysis, then we'll describe fermentation, and then progress to aerobic and anaerobic respiration. We'll do just a little bit of discussion on photoautotrophs and oxygenic photosynthesis, and then we'll finish with a look at the nitrogen cycle and how microbes are involved in the nitrogen cycle. So this is kind of our worldview, looking at different ways that chemoheterotrophs can produce ATP, and you'll recall that ATP is the so-called energy currency of the cell. It's used in many processes that require extra energy, cellular work such as anabolism, biosynthesis, active transport, and motility. And since the cell is constantly using up ATP, the cell has to constantly be making ATP. So we'll take a historical approach to this. We'll back up in time to 3.6, 3.8 billion years ago when the first cells evolved and see how they may have produced ATP. So you'll recall that the first cells, the first prokaryotic cells, arose about 3.8 to 3.8 billion years ago. Conditions on Earth were really different from today. The atmosphere was anaerobic, meaning there was no free molecular oxygen present. And it wasn't until the evolution of chlorophyll A and cyanobacteria, and there's some debate on when that occurred, but with the evolution of chlorophyll A by cyanobacteria, they started pumping out molecular oxygen into the atmosphere, and slowly, the Earth's atmosphere became aerobic. Today we have an oxygen concentration in atmospheric air of about 21%. But again, we just want to emphasize that when the first cells evolved, there was no molecular oxygen present in the air. Also, electron transport chains hadn't evolved. So the cells could not carry out the process that we humans are most familiar with, which is aerobic respiration. So these primitive cells, they lacked electron transport chains. There was no molecular oxygen, so they had to find another way of making ATP. So the first strategy we believe that evolved was a relatively simple way of making a little bit of ATP, starting with one glucose molecule. And this strategy is called glycolysis or glycolysis. Literally, it means sugar splitting. And we'll see that this is a way to use the energy in glucose and drive a little bit of ATP synthesis. So let's first take a look at this primitive way of making ATP. So this is a so-called Embden-Meyerhof glycolytic pathway. And again, this is the kind of the big view, the world view. You do not need to memorize each step. You don't have to know the enzymes involved. What we want to really focus on are what are the main events. And down in the left-hand corner there you can see some of the main events. So big events are going to be, we're going to take our 6-carbon glucose and we're going to split it into two 3-carbon end products called pyruvate or pyruvic acid. During this 10-step process, it requires 10 different enzymes. Some of the high-energy electrons will be stripped from glucose, and these high-energy electrons will be transferred to the electron hydrogen atom carrier NAD, So we're going to end up with 2 reduced NADH. Through the process called substrate level phosphorylation, we're going to make a total of 4 ATP. But to get this whole process started, the cell has to use 2 ATP. So we say the net gain of ATP for the cell is 2 ATP per glucose in glycolysis. Okay, and again, this is just the key points that you would want to remember for glycolysis for our next lecture exam. Now, we use the phrase substrate level phosphorylation is how ATP is made during glycolysis. And substrate level phosphorylation is when a phosphate group is transferred from one organic molecule to another. So in this case, substrate level phosphorylation, you'll see that we're going to transfer a phosphate group from the molecule that's being held by the enzyme, and it's going to be transferred to ADP to make ATP. We will see that this is really different from the process of oxidative phosphorylation that we'll see later when we start talking about respiration, in which a proton gradient is formed across a membrane by an electron transport chain. And then that proton gradient will be used to drive massive ATP synthesis by a cool enzyme complex called ATP synthase. So again, we want to make sure that we're feeling confident with the main events in glycolysis. And now we want to look at some of the problems with glycolysis. So you'll recall we said that during glycolysis, high energy electrons from glucose will be transferred to NAD to form 2 NADH, our reduced forms. The problem is cells have limited amount of oxidized NAD. And if they were to use all the NAD up during glycolysis, then glycolysis will stop. And if you take a look at our map here, you'll see that ATP synthesis occurs in the steps following the reduction of NAD to NADH. So if the cell uses up all of its NAD, glycolysis will stop, and the cell can no longer make ATP. And of course, that would be deadly for the cell. So those primitive cells had to find a solution. How could they take the reduced NADH and oxidize it, right, to keep a continual supply of NAD? In the early strategies for this process, the cells evolve fermentation. So here's 2 examples of fermentation that it's important that you know for the lecture exam. And the first one to the left here is the final steps in what's called lactic acid fermentation. Many of the fermentation pathways, they're named after important end products. So we can see here that in lactic acid fermentation, the pyruvic acid or pyruvate, which is one end product of glycolysis, is going to be combined with another end product of glycolysis, the NADH. The NADH will transfer its hydrogen and electrons to pyruvic acid, oxidizing the NADH back to NAD, and it's the oxidized form of NAD that will keep glycolysis operating, keep ATP synthesis continuing. And we'll see then that another important end product of lactic acid fermentation is lactic acid. The second fermentation pathway we'd like you to know is called alcoholic fermentation. This is a little bit more complicated. You can see that in alcoholic fermentation, again, we're going to start with our pyruvic acid, our end product of glycolysis. We're going to tear off one of the carbons and release it as carbon dioxide. So that's a carbon dioxide that causes our bread dough to rise. It's carbon dioxide that can add the carbonation, the sparkling, to sparkling wines, for example. We end up then with what's called an intermediate, a 2-carbon acetaldehyde, and it's the acetaldehyde that will act as what we call the terminal electron acceptor. That is, the reduced NADH is going to pass its electrons, its hydrogen atom, to acetaldehyde. Again, the goal is to get that oxidized NAD to keep glycolysis operating. And then once we reduce the acetaldehyde, we're going to end up with our 2-carbon ethanol. So here's the alcohol of alcoholic fermentation. So in lecture, we'll be talking about how humans have used microbial fermentation to their advantages. With lactic acid fermentation, again, we can preserve foods in the absence of refrigeration. Lactic acid fermentation of vegetables, for example, to make sauerkraut or kimchi. This is a way to preserve the food in the absence of refrigeration. the acids produced will drop the pH, and that will inhibit the growth of spoilage microbes. In addition, there are lactic acid bacteria that colonize our mucous membranes, and there they take sugars our bodies produce, ferment them to lactic acids, drops the pH, and that low pH is going to inhibit the growth of opportunistic pathogens such as the yeast Candida albicans. It's believed that many good members of the human microbiome are lactic acid bacteria, and many of the so-called probiotics contain lots and lots of lactic acid bacteria. With the alcoholic fermentation, we've discussed how the yeast, the baker's or brewer's yeast, Saccharomyces cerevisiae, performs the alcoholic fermentation. Again, we could say that it's through alcoholic fermentation that the nutritive value of grains and, say, grapes could be preserved. Again, the alcohol, the ethanol, will inhibit the growth of spoilage microbes. And indeed, probably historically in Europe, at many times it was probably safer to drink beer or wine than it was to drink regular drinking water, because the drinking water so often was contaminated with feces. So you'd have fecal-oral transmission of intestinal pathogens. And again, the alcohol would help to inhibit or kill those potential pathogens. Oh, I now forgot one more example of lactic acid fermentation. If we take milk, dairy products, and we carry out lactic acid fermentation, we end up with buttermilk, kefir, yogurts, cheese, and again, this is a way to preserve the nutritive value of the milk in the absence of refrigeration. So fermentation was first described by Louis Pasteur, and he described it as life in the absence of air, life in the absence of oxygen. So here are some key points that we'd like you to know about fermentation. So it's the partial oxidation of glucose, meaning we're not stripping off all the high energy electrons. The pyruvic acid or the fermentation products, for example, the lactic acid or alcohol, still have lots of high energy electrons. So we could argue it's a little bit wasteful. We talk about terminal electron acceptors, that is, the electrons that were originally stripped from what used to be glucose, where are they going to end up? And in the case of fermentation, it's always a so-called internal organic electron acceptor. Internal meaning it's a molecule made within the cell, and organic meaning it is organic. So in lactic acid fermentation, the terminal electron acceptor would be pyruvate or pyruvic acid. In alcoholic fermentation, it would be that intermediate acetaldehyde. Notice that fermentation does not require oxygen, so that was really important when the first cells evolved. So it's an anaerobic process. It also does not require an electron transport chain, and you'll recall ETCs had not yet evolved when those first cells did. So total gains for the cell would be 2 ATP per glucose via substrate level phosphorylation. And again, those waste products, the lactic acid, for example, the alcohol, they have lots of high energy electrons. So we could say the waste products, end products, are energy rich. So that this is rather a wasteful process. And even though we've emphasized how the fermentation end products, the acids and alcohols, can help inhibit the growth of spoilage microbes and thus can help preserve foods in the absence of refrigeration, it's important to note that those acids and alcohols, if they build up, can actually be toxic for the cells. This is just an image so that we can see that there's a number of different fermentation pathways in the microbial world. Different enzymes would permit microbes to carry out different fermentation products. So we can see here, starting with the pyruvic acid, we've discussed the lactic acid fermentation carried out by the so-called lactic acid bacteria, which includes members of the genus Streptococcus and Lactobacillus, and then some members of the genus Bacillus can carry that out. We've described the alcoholic fermentation carried out by Saccharomyces, and then there's some additional fermentation pathways, folks. You need not memorize the additional pathways for the lecture exam. We will come back in lab later in the semester and talk about the mixed acid fermentation carried out by Escherichia coli, E. coli, and Salmonella. We have a diagnostic test so that we can detect bacteria that carry out mixed acid fermentation. We'll learn about that in lab. And then a close cousin of E. coli, Enterobacter, carries out a different fermentation process called 2,3-butanediol fermentation. And again, we'll learn how to detect that 2,3-butanediol fermentation process in lab. And again, this is just showing how humans have taken advantage of microbial fermentation products to make fermented milk products such as cheese, yogurt, soy sauce is a fermentation product, wine and beer, nail polish remover and rubbing alcohol, not yummy, don't eat those, you guys. And fermentation is involved in the production of vinegar. And again, just some more human uses for fermentation. The one we want you to remember again is lactic acid and alcoholic fermentation. Oh, and very importantly, we want to remember that in the production of alcohol, not alcohol, sorry, the production of chocolate, chocolate involves fermentation steps. So if you like chocolate, those microbial fermenters are important in the chocolate production process. And again, we're going to be learning how to detect fermentation in the lab, and these will be really important diagnostic tests that we run in the lab to identify unknown bacteria. Okay, so what we'll do is we'll just end this little discussion on fermentation with problems with fermentation. So you'll recall that the end products still carry high energy electrons, so it's a very wasteful process for the cell. And the end products, the acids and alcohols, can actually be toxic for the cells if they build up. So because fermentation is wasteful, we could say that after maybe many, many years as the cells grew and divided, they may have encountered a food shortage because they were wastefully using up all of their preformed organic molecules. So there would have been natural selection for mutants who could make more ATP per glucose without making such toxic end products. So it was these mutants that were selected that eventually evolved aerobic respiration. So we're going to end this video here, and then we'll be introducing aerobic respiration in the next video.